one of the disadvantages of Zoom is it's a lot harder to make a talk interactive. You know, normally for this kind of talk, it would be very useful for you to be able to stop me at various points and ask questions. It's quite difficult to do that with Zoom. So what I'll probably do is, because I'm going to talk to you about the two fundamentally different types of stem cells. So I'll probably stop halfway. But we can take if there's any points at that point, and then I'll go on to the second part. I'll try not to make it too long or too detailed, but at the same time, there's lots of really interesting and important information that I'd like to get across. So, you know, yeah, you don't need to know all the details. I just really want to give you a flavor of why this is such an important and exciting area. Okay, so let's hope the share screen will work.
I think that sounds absolutely brilliant. And I just before we make a start, I'd just like to say it is a great pleasure for you to thank you so much for coming along, so Austin Smith, and just sort of let the others know just that your main role at the moment is Director of the Living Systems Institute up at the university, as well as having this sort of extensive track record in stem cell research. So it is a great pleasure that you've been able to come and talk to us today.
Oh, thank you. You know, it's I'm very pleased to do so. I've worked on stem cells actually all of my research career, and I've worked on the type of stem cells we're going to get to at the end of this talk. First, I'll give you a more general introduction. So this is a newt, and it's had its arm chopped off. And the remarkable thing that happens in these animals is that you can see that over about 2 weeks, this arm will regrow itself, or it's a leg actually, and it will regrow perfectly to exactly the same size as the original. And you can do this actually with many different parts of the body of these animals and of many other animals. And the reason this is possible is because of the stem cells in these animals that have the potential to reform new structures. So this is a kind of the most dramatic example, really, of the power of stem cells. In mammals, and in particular in humans, we don't have the regenerative capacity on such a dramatic scale. But stem cells are actually in some ways even more important just to keep us alive. So I'm trying to illustrate this here with the example of the blood. So every one of us, every day, has to make this very large number, 100,000 million, 10 to the 11, new blood cells, just to keep us alive. So that's 10 to the 15 in our lifetime, kilograms and kilograms of new blood cells. Because most of the cells in our blood system are very specialized, they perform a specific function, and they die quite quickly. So we constantly have to make new blood cells. It's not just the blood. The entire intestine turns over every week. The entire skin surface every 2 weeks, constantly new cells. And in males, billions of sperm cells are produced throughout life. So we're constantly having to make new cells and reconstitute our tissues. And this relies on stem cells. So the special thing about stem cells is that when a stem cell divides to make two daughter cells, two things can happen. It can make an identical copy of itself, and that's a process we call self-renewal. And that's really the defining property of a stem cell, self-renewal. or it can differentiate. And that means it can start to become a more specialized type of cell. And the critical thing is that this process of self-renewal in a true stem cell will go on throughout the life of the animal. So what that means is the stem cell always survives, and it always is making the differentiated cells, and they'll go and perform their job, and then they may die, but they can always be replaced because the stem cells are always surviving. So that's why they are so important and so special. So this is a bit of a busy slide, but what I'm trying to show you here on the left-hand side, this example is the blood system. And on the left-hand side, we have the cartoon of the stem cell. And that cell can self-renew itself. It lives in our bone marrow, or it can differentiate. And when it differentiates, starts to differentiate, it makes what we call progenitor cells. So those cells divide a lot. And every time they divide, they become a bit more specialized. So you end up with a range of different specialized cells, still multiplying. So you're making very large numbers of them. But this is a conveyor belt. They're all moving then in one direction, which is towards the final functional differentiated cell, which can be the immune cells, it can be the platelet producing cells, it can be the red blood cells. So the point is that these cells at the end usually cannot divide anymore, and often they live for a very short time, sometimes only hours. So they have to be constantly made through this conveyor belt system. And as long as we have the stem cell, we can continue to make new blood. So these stem cells are actually quite small in number, and they're not dividing very much. And that's a protective mechanism because there's less risk of them getting a mutation and becoming cancerous. So the blood stem cells are the first example, and still the most wide clinical example of using stem cells in a stem cell transplantation, because blood stem cells can be transplanted from one person to another. And so this is used to treat different types of leukemia. And nowadays it's combined with gene therapy also to treat some genetic immune diseases. So basically you replace the patient's blood system entirely with the blood system from another person by transferring their stem cells, and then the introduced stem cells will take over. The limitation with this approach, which is a life-saving therapy used throughout the world, but it's never the first approach in the clinic because it is risky, because you're having to use stem cells from another person. So even if they might be a sibling, they're never going to be identical. And so there's always a risk of immunological reaction. And one of the limitations we have is that we can't grow blood stem cells outside of the body, despite many, many years of attempting to do so. So there's also always a limited number of them that we're able to transfer. But still, this is the most common use of stem cells in the clinic. So another type of stem cell that was characterized at the same time as the blood stem cell in the 1970s and the 1980s is stem cells from the skin. So what I'm showing you here, this is a Petri dish, and these little blobs here, these are actually colonies of a few hundred cells, each of which has grown up from one cell. And they'll keep growing in the dish, and eventually they'll cover the whole dish. And when they do that, they make a sheet of skin. So here, this sheet of skin has been detached from the dish, and you can see that this has it's a tensile structure like normal skin. And what you can do with that is you can transplant it to people who need a skin graft. So this whole field was pioneered by Howard Green and colleagues, and they took this into the clinic in the early 1980s in the case of 2 boys who'd had very serious chemical burns. And this shows you, these are the sheets of skin that have been grown in the laboratory and are then placed onto the area of damage where there is no normal skin left. So this is only necessary when there's a very serious and widespread damage to the skin. It's actually most commonly used in the case now of military incidents. Because normally for a small injury, our own skin stem cells are sufficient to be able to regrow. But if there's a massive loss, they can't do that. But in this case, what you do is you're still salvaging some skin stem cells from the person themselves that you grow up in the laboratory to make new skin. And then it can be transplanted and there won't be any rejection. And very recently, this type of skin transplantation has also been combined with gene therapy to treat severe blistering condition called junctional epidermolysis bullosa, where the children with this disease, their skin literally falls off if there's any kind of physical contact. And we now know the causes of this, the genes, and so it's possible by gene therapy to put in a rescue gene into the stem cells in culture, and then to grow that up in skin in the laboratory and then transplant that skin. So the next slide, I have to warn you, if you're a bit squeamish, you can look away because I'm going to show the slide of a boy in the hospital, really almost at the end of life. This is the patient here, and then this is after the skin that has been grown up in the laboratory. It's had the rescue gene introduced, and then you can see the skin grows up after 1 month, and then after 4 months, although this doesn't look perfect, this is actually pretty good skin, and these little blisters that are left actually covered over in the subsequent months, so that this boy was able to leave the hospital and live a relatively normal life. So again, this is a pretty spectacular life-saving therapy, albeit for, let me just stop that, albeit for relatively rare diseases. But for a long time, we only really knew about these 2 types of stem cell, the skin stem cell and the blood stem cell. But recently, in the last 10 years or so, we've found out a lot more about stem cells in other tissues and organs. And we've also found a way to grow them in the laboratory. And this is called organoid cultures. And so what this movie is showing you is a structure that's grown up actually originally from 1 stem cell from the intestine. I'll just restart it again. You can see it grows up in this kind of cyst ball, and then it branches off these structures. And this will keep growing like this. And we can now make these structures from many different organs, the colon, the stomach, the liver, the pancreas. And when we examine them, we can see that they contain different types of specialized cell. And they're really very similar in many ways to the normal organ, in this case, the intestine. So this is very interesting. How useful is it medically? Well, there's a huge interest now in using organoid systems, in particular in the study of cancer, where you can compare the normal tissue with the cancer tissue. You can also use this system to understand exactly what type of cancer the individual might have, and you can use them to investigate the mechanisms of the cancer and possible treatments and new drugs. You can also use organoids for personalized medicine from patients who have particular types of disease. And again, you can test drugs directly on the cells, on the tissue from the individual. In cases where, which is often the case, there are many drugs available, but the individual patient will only respond to a particular drug, and either you have to try it on the patient themselves, which is going to be very expensive and time consuming, or you can now test the drugs in the laboratory on these organoids made from the person and work out which drug is going to work. But with the organoids, it's still in the future to be able to use them for transplantation for tissues like the intestine and the stomach. Although there's, of course, a lot of research going on into that. So, adult tissue stem cells, their job is to maintain individual tissues, to renew them throughout our life. And I've listed here the main places where we find them. But there's two important things to be aware of. These stem cells are dedicated to making the cells of the tissue that they live in. So they don't make cell types of other tissue. So the blood stem cell can't make skin, and the skin stem cell can't make blood or brain. So they're very good at renewing the tissue that they belong to. And they won't make mistakes and make the wrong type of cell. But of course, that limits them also. And furthermore, we don't have stem cells in all of our tissues, because some tissues, like the brain or the heart, they don't go and undergo any renewal. So there's very little cell division in the brain or in the heart after early life. So again, we don't have ways of studying or repairing those tissues using tissue stem cells, because there isn't a relevant tissue stem cell. So I'm going to stop for a moment here and ask if anyone has any questions or comments, if anything is unclear or confusing on this first part. The only problem is I can't see, but maybe if somebody's chairing, they can tell me if there's a...
I can see Angela Shaw has got her hand up at the moment. So Angela, if you'd like to ask or comment.
Austin, thank you. That was so clear. Can I just ask you one question about the mini?
Oh, I've lost you, Angela.
About the mini intestine that you showed.
Yes. Yeah.
So, do I understand correctly that that one cell that you'd taken from the intestine was able to grow into all of the different types of cells in the intestine?
For the purposes of simplification,
yes.
Yes. I mean, it doesn't have, so it has all of the epithelial cell types of the intestine. Of course, it doesn't have the blood cells or the nerve cells that would normally. And there's a lot of interest now in making more complex structures that really have everything. But it does make all the cell types of the intestine. And then what happens at the end of the movie there, you basically just break this up into small pieces, and then they'll keep growing and make more and more. So they can just keep on going forever. And of course, you can use that then to study what's the difference between a normal patient's intestinal organoid and the intestinal organoids made from people who have intestinal cancer, for example.
Yeah, thank you.
And I see, Robin, you've got your hand up, and then I see John's got his hand up after it as well.
Thank you. Yeah, Austin, I was fascinated by the growth of the skin's stem cells into new skin. What is there a time scale that allows for so many square centimeters of skin to be grown? What is it?
Yes, so it's actually remarkable, because you have obviously to transplant a whole an adult patient, you need square meters of skin.
Yeah.
But in fact, that can be done. They take a biopsy of around about 2 centimeters square. So usually that will be from the groin or under the arm, somewhere that hasn't been destroyed by the burns injury. And in about 3 weeks, because these cells grow so fast, it's possible to grow up enough skin then to do the transplant. They may do 2 rounds of transplant, because it may be too much to graft.
Yes.
Just surgically, actually, not so much
imagine.
to be able to do like both front and back at the same time. But it actually happens very quick, and it has to. If it took too long, then you wouldn't be able to keep the patient alive without skin. You know, they're very, very vulnerable in that
of course.
critical curve. But fortunately, it happens very fast. It is expensive to do, because it has to be done under very carefully controlled regulatory conditions.
Of course.
Different from how we would do in an experimental.
No. Puts me in mind of an old TV series, something along the lines of, we have the technology, we can rebuild him.
Well, that is sort of is the dream of stem cells. But like I'm saying on this last slide, it's not true. There are some incredible examples, but it's not true for lots of us. We're not like a newt. We can't regenerate.
Well, I think I know a few newts, but no, thank you very much. That's fascinating.
Then I think it's John, followed by Narina.
Austin, thank you so much for such clarity. When you want to find an adult tissue stem cell, how do you identify the stem cell in the other cells in that tissue?
Well, you see, that's what I lecture the students on, and that's a whole 1-hour lecture just on that question. It's actually there are a variety of different ways of doing this, because these cells are usually quite rare, and in some cases we're still searching for them. Typically, we use a combination of some kind of an assay system. So for the blood cell, you can use transplantation. So we can make the mice very sick by destroying their blood system, and they'll only survive if we give them new stem cells. And so you can use that to work out which cells in a population are the stem cells, because only the stem cells will be able to keep the mouse alive. And we have different kinds of technologies to sort out between different cells and eventually work out, almost by a process of elimination.
There's no simple morphological difference you can identify.
There's not. I mean, once you know which cell is the stem cells, sometimes morphology can help you, but not in advance. We also nowadays, because we have the genome sequence and we can look at all the genes that are expressed by cells, then we can use gene markers to help us in this search to identify which cells are the stem cells. But again, you always have to, there's no marker code alone is sufficient. You have to have some functional tests to demonstrate that is the stem cell.
Well, I don't want to hold the time. Suffice it to say, it isn't just like falling off a log. There's quite a lot involved there.
It is, and I should say it's almost always done first in the mouse, where we can do these experiments. It's actually a very important instance of where experiments in animals are
Yeah.
critical. Almost everything has started in mouse and then gone to human in the stem cell field.
Thank you.
Okay. And then Narina, your question.
Thank you. Thank you for devoting your life to this wonderful research, Austin. When I was a little lass, around about 1946, I think I must have been somewhere near Archie McIndoe's operations. I used to come across a lot of severely burned people who had been treated. I take it with this new stem cells for skin that they won't look so horrendous in future and much more, what I might say, normal. Is that correct?
So, I mean, there are, of course, a lot of other improvements in plastic surgery and in skin grafting. Very often now, they can graft skin from one part of your body to another. So, which is still reliant on the stem cells for the new graft to take. But it's only in the extreme case where really almost all of your skin is gone, which is why, for example, very serious burns, or as I say, most commonly now in a military situation, where it would be so severe that the only way you could get enough skin would be to grow it in the laboratory. So it's not a routine thing, but it's definitely life-saving for those people. I have to say, the skin, one of the issues also faced is that the skin that is regenerated in this way, it doesn't have sweat glands or produce hair, so it's not completely normal skin. Again, the reasons for that are believed to be because of the damage of the burn has killed off the support cells that would normally allow the hair and the sweat glands to form, because they are part of the normal skin. So that is an ongoing disadvantage for people who are treated in this way, and then is another area of research to try and improve that situation.
I used to, as a little lass, I used to see these men.
Oh, yeah, it's just all.
Yes.
It looks so painful. It looks so tight.
Yes. Yes. I mean, I think with again, they can, I believe, do that a lot better these days with skin grafts rather than just allowing the skin to regrow itself. Yeah, I mean, there is, as I say, there's a remarkable capacity of these cells, but sometimes, because the natural healing process is, of course, always focused on closing the wound as fast as possible, but that's then what can lead to these scars and constrictions. So nowadays, the plastic surgeons are very sophisticated in how they can manipulate that to get a better healing process and a better graft. Yeah, but again, for the long-term renewal of that skin, there has to be the stem cells in the graft, otherwise it will die away within weeks.
Well, thank you, and thank you for the memory of Archie McIndoe and the previous pioneers of this wonderful subject. Thank you very much, Austin.
Is there any more questions at the moment? Okay, I think there's no more.
Okay, so now I'll move on to the second part of the talk, where I'm going to talk about another type of stem cell, which isn't involved in keeping us alive at all. In fact, this is a stem cell that really only exists in the laboratory. It's actually the type of stem cell I work with myself, and I hope I'll convince you why it's interesting and also has great potential in medicine. So, as I've just told you, the adult stem cells are highly specialized in what they can make. And so, ideally, one might want to have a cell that could make every other type of cell. And the word we use to describe this is pluripotency. So one cell that can make every other type of cell in our bodies. And actually we know that there is no cell like that in the adult or after birth in any mammal. There are in some very simple organisms like flatworms, but not in mammals. But of course, we all came ultimately from one cell. And we know that in the very early embryo, there are cells that have this property of pluripotency that have the potential to make every other type of cell. So back in 1981, again, in experiments in mice, groups in Cambridge and in San Francisco took very early mouse embryos that are called blastocysts. So they've got, at this stage, they've got about 100 cells in the embryo. It hasn't yet fixed into the uterus, so it's still a free-floating structure, so quite easy to get out from the mice. And they know that within that blastocyst, there's a group of cells that are pluripotent because they're the cells that are going to make the whole embryo. And so they put them into the Petri dish in different types of conditions. And they found a condition that would allow these cells to keep growing. And then when they test these cells, so they grow from single cells into these colonies again of a couple of hundred cells here. And these cells, you might think, well, they don't look very interesting. They're very small cells. They all look the same. And they'll actually just keep growing like this forever. But the interesting thing is they look like the cells in the early embryo. If you change the culture condition, the environment they're in, so you give them some different stimuli, then they'll start to make all different kinds of cells. So they'll make nerve cells, they'll make heart cells. You can see here spontaneously contracting. Here they're making pancreatic type cells. So they can make every type of cell. And now we know that these cells are actually almost identical to cells in the very early embryo because we can look at all the genes they express and it's the same as we see in the early embryo. But unlike the cells in the embryo that only stay in this state for 1 or 2 days, these cells, the embryonic stem cells, if we keep them in the condition for self-renewal, can stay like that forever. And then if we change that condition, they're always able to differentiate. So this has been an incredibly powerful research system in the mouse. And we know lots now about how these cells are regulated and which genes are important. I'll come back to that in a minute. But of course, you can't use a mouse to treat any kind of human disease. And it took a while, nearly 20 years, before people succeeded in making similar types of cells from human embryos. So this is a human blastocyst, which is very similar to the mouse, a little bit bigger, probably about 200 cells. And again, inside of this structure, there's a small group of around 20 cells that are the pluripotent cells that would make the human embryo. And this population of cells, if we put them in the laboratory into culture, again in a very specific condition, it's been worked out by trial and error really, then you can keep them growing. And like the mouse embryonic stem cells, they'll keep growing without differentiating. But if you change the culture condition, they can make all types of differentiated human cell. So this is again, very exciting. I should just explain where this embryo comes from, because we don't take human embryos out of women. These are embryos that are generated for in vitro fertilization. So they're generated in the laboratory by test tube fertilization. And then if they're not needed anymore for a pregnancy, then they can be donated for research. That in many countries has been quite controversial, still is in many places. But in the United Kingdom, that was the possibilities of that type of research were foreseen by scientists in the 1980s, and so it was written into the law in the UK that you could do research in those circumstances on human embryos. But nonetheless, that it made it quite controversial, human embryonic stem cells, and held back the research for quite a while, even though the medical possibilities are quite clear for these cells. But a great breakthrough came a few years later from this man, Shinya Yamanaka, a Japanese stem cell researcher. And what he did was he took the knowledge we'd gained from studying the mouse embryonic stem cells and identifying the important genes for pluripotency. And then he had the idea that if he put those genes into an adult cell, they might change that cell back to the pluripotent state. So this is something called molecular reprogramming. And he succeeded in that. So he took skin cells from adult mice and he introduced 4 genes from the embryonic stem cells. And then he found if he put those cells into the culture condition for embryonic stem cells, the cells would change from being a skin cell into being a pluripotent cell, actually identical to a cell derived directly from the early embryo. So this is something that never happens in normal life, in the normal animal. This is a laboratory procedure, but remarkably, the cells it gives at the end are pretty much perfect. And not every time, but if you're careful about how you do it and how you test the cells, then often they will be indistinguishable from cells you took directly from an embryo, and they can do all the same things. So Shinya, again, quite remarkably, was able to move in 1 year from demonstrating this in mouse to show he could do the same thing in human. So now we can make human pluripotent stem cells without needing an embryo. So we can have human pluripotent stem cells either from embryos or by molecular reprogramming from individuals, which may be patients. And what we can do with them is we can differentiate them into many different types of cell, and depending what tissue we're interested in for research or for disease, we can make really any type of cell from them. And in this case, let's assume we're interested in brain cells. And then what we can do with them is really 3 different types of things. We can use them as disease models to study what goes wrong if we've taken these cells from a patient who has a disease like motor neuron disease or Alzheimer's disease. And of course, you can't take the cells out of the brains of those people. So we've never really been able to study this in the past, except in autopsy samples. But now we can take a bit of skin or a bit of blood from those people, create the induced pluripotent stem cells, and then differentiate them into the nerve cells or brain cells, and then examine why are they different, how are they different, and why from the same cells made from a normal healthy individual. And we can find out the genes that are involved in this. And then we can move on from that to use these same cells to test drugs that we may have developed candidates by knowing a bit more about the disease process. And again, because we can grow huge numbers of these cells, we can do screens of thousands or even tens of thousands of drugs to try and find drugs that might reduce the symptoms or even cure the disease. And this is really now the main use of human pluripotent stem cells. But the big dream, of course, is that you could use them for transplantation therapy. And in particular, the induced pluripotent stem cells, in principle, would have the huge advantage that because they come from the patient themselves, then you wouldn't have an immunological rejection problem. This is still in its infancy, and it's also very expensive. And actually, I think that's going to be a permanent problem. And that whenever you can find a drug treatment, it's always going to be cheaper than having to do a cell replacement therapy. But there are clinical trials at the moment, possibly may have heard about them, for age-related macular degeneration. And for Parkinson's disease, there are trials for both of these in the UK, as well as in America and Japan. And there are also trials for in type 1 diabetes to replace the insulin-producing beta cells. So, you know, this is also an area that is moving into the clinic, and there's actually big issues that people are beginning to think seriously about now, about how to make this an affordable treatment in future. And then the last thing I want to show is just an example of the kind of things people are beginning to do now with human pluripotent stem cells that actually stimulated by what people have been able to do with the adult stem cells with organoids. Because what people are now doing with human pluripotent stem cells is trying to organize, control the differentiation in the laboratory so that they begin to make micro tissues. And the most perhaps exciting example of this is in the brain, where we can, if we do the differentiation in the right way, then the cells will organize themselves into structures that are like parts of the developing human brain. And you can see here the kind of complex organization that we get that does look like the brain of a human fetus in the uterus. And again, this, you know, has huge potential for modeling disease, for perhaps beginning to understand things like autism, and again, for testing drugs. Of course, you know, it might also be considered to be a bit scary, but these are not brains that can think or have any sensation. But still, actually, that is something that, you know, people debate quite a lot, and how do we ensure what safeguards do we need in place in this area? Okay, I think that's my last slide. So I'd be very happy now to take. If I stop sharing, I can see everybody, and we can discuss.
Okay. Okay, can I say something? Number one, thank you so much. I'm so glad your colleagues with you, because I think you modelled so well. Number one, the pace of what you were saying, and that idea of stopping in the middle, I think is really valuable. I found it very valuable. And also the ratio of talk to slides. The slides were very clear, and there wasn't a lot to read on them. You know, you were using diagrams a lot. It's just so much easier when you're listening and looking at the same time. So I thought the presentation was excellent. Thank you.
Oh, thank you very much, because you know, that's great to get that feedback, because we never quite know.
No.
Yeah, and actually students never give you that kind of feedback.
Well, no, but, you know, listening and seeing at the same time is difficult in terms of
Yes.
interpretation and processing. But I think you got the balance. For me, you got the balance just right there. And of course your material was just really exciting. And
yeah, well, of course it's easy to talk about, because it is cool.
That's right. And it's got such implications, hopefully, for, you know, future work. But there were two things. These are just quick questions, hopefully. Number one, you talked about the human embryo, using human embryos, and how, you know, we've got legislation to cope with that in this country. But is there an age limit? That the embryo that we saw was 6 days old. Presumably there's a limit. I mean, would you be able to use an embryo that was, you know, a month old, something like that?
Okay, so there's two things there. The embryo I showed at day 5, day 6, day 7. That's the stage when the embryo has the pluripotent cells we're interested in. So, and actually, then on day 7, day 8, the human embryo implants into the uterus. So embryos are never available after that stage.
Okay, that's
so they're only maintained in the IVF clinics to day 5.
right.
That's when they would transfer them to try to get a pregnancy.
That's
But one of the other aspect is that the law says, so we could continue to try to grow them intact in the laboratory, and that's partly what my colleague will talk about in a few months' time. Because that's also very interesting and important, because we have no knowledge really of how that early human embryo develops once it's implanted. But currently the law says you can only keep a human embryo alive for 14 days.
Right. Thank you.
Which, yeah, we can leave it there, because she will talk more about those aspects.
Yeah, on the ethical issues involved. The other one, hopefully another quick question. Right at the very beginning, you said your entire intestine is replaced every week, which that's quite refreshing, really, doesn't it?
Yes, yeah, because these cells are working very hard, and of course they're also getting battered all the time by the.
Well, that's right. For the work they have to do.
Yeah, exactly.
So, this might be a naive question, but given that there's so much, for example, inflammatory bowel disease, and people suffer with ulcers and other injuries in the intestine, why is it that replenishment doesn't mean that the intestine is then disease-free?
That's a good question. I mean, in the case of ulcers, serious ulcers, that is one of the targets why people are interested in the potential of a replacement therapy, in the end being able to use organoids to put a graft in over the ulcer. And there's even been some tests of principle of that in mice. Again, it's not my area. I think there are the ongoing inflammation issues are one of the reasons why basically the resident stem cells are not able to get into these areas where you have the ulcers. I honestly don't know to be able to say anything informed about conditions like inflammatory bowel disease.
Can I get you off the hook by changing the subject quickly? I'll take my hand down now. Hello, Austin. Charlie Lane here. A layman, absolutely, but I do agree with a hugely well presented subject, and that's important because it's complex and could just remain complex. So for this panel without the medical background, who have to make judgments on studies, I'm intrigued to know whether or not you have applied or plan to apply, or your colleagues, to the bank in the context of muscular reprogramming, because 10,000 people exist there. Are you likely to ask for samples of whatever type of cell? Are we in the same parish here? Help me make that leap.
I'm not quite sure what bank you're referring to.
The Exeter 10,000 bank.
Okay.
So Austin, if we just fill you in, so our colleagues here will help us to decide whether a project coming into the Exeter 10,000 is suitable for the volunteers in Exeter 10,000 to either come in and donate something, or to use their data or genes or samples which we have stored. So I think Charlie's asking you if it was likely that you might need some samples from healthy controls or from patients with X, Y, or Z to help you take adult cells and use them, or perhaps even to convert them into pluripotent cells.
Okay.
Thank you, Angela. I was exactly asking that.
But you mentioned muscle. Is it specifically for muscle, or could it be for any disease?
Anything.
Could be, yeah.
Anything.
So actually, there was something then. I think this is similar to the a big project in Cambridge, where they had established a big bank of cells and of patients from which they made many of these iPS cells. So my own research group is not directly involved in that type of research, and we currently don't have anybody in the institute who is, but I know there is at least one group in the medical school. I'm thinking of John Mill, and there are plans to recruit more researchers, both in the medical school and potentially in this institute, for whom that type of resource might be of interest. I can only say might because it depends exactly what the area of research is people are doing. Sometimes, so we have a group in our institute, for example, that works on motor neuron disease. It's a very serious disease, using iPS cell models. But he has to go, because the patients are relatively rare, he has to go specifically to repositories for those patients rather than a general collection. So and that's not I can't give you a precise answer because it really depends on the particular researcher.
And we know that John Mills is already working with the Exeter 10,000 with requests, so we've already got that relationship and rapport with John, which is great. Is there any other questions a moment or comments? Angela. Sorry.
Can I just ask one for clarification, Austin? I've probably turned away and missed it. So when you take your adult cells and turn them back into embryonic cells, from the mouse studies that you showed was the first one, they introduced 4 genes. Mm-hmm. So is that what you do in humans as well?
Yeah, it's exactly the same, but now we can do it. The technique we use in our laboratory, we introduce the RNAs, not the genes. So, okay. the advantage of that is that there's no permanent genetic
Yeah,
change.
yeah, that makes sense.
And so this is actually really, really nice technique and seems to work very, very well.
Yeah, very good. Thank you. Brilliant.
And Narina, have you got your hand up? Yeah.
Yes. Any chance of you coming back in a year or so and tell us the progress that's been made?
well, you're going to hear from my colleague in a few months. She'll actually talk about a slightly different aspect about the embryo cell and how we can study that. And then, yeah, I think, yeah.
And what
a year
Austin,
is a short time
Austin,
in research.
Yes, exactly. That's what I was going to say. What Austin was describing to you is probably 20 years worth of research, not a single year. So,
not 20 years. Yeah, I do agree. It was a wonderful presentation. I've lost count of the number of really expert lecturers who've put slides up and read from them. And I feel like putting my hand up and saying, I went to school, I learned how to read.
Yeah.
No, it was first rate.
Thank you.
Is there any other questions a moment? Okay, I think that was just an absolutely fantastic talk, Austin. That was absolutely brilliant. So thank you ever so much. And I know some of our representatives who are unable to make it today will be looking at the recording. So that's absolutely brilliant to have that resource as well. And we really look forward to hearing the next instalment in November at the Lay Day.
Yeah.
Thank you.
So thank you so much.
Thank you.
The only other thing I was going to just say is on behalf of the representatives, I hope it's okay, everybody. But I think the other thing that we can also provide is the Exeter 10,000 lay group is actually sometimes if you need to get sort of the public or patient point of view on any sort of future grant applications, that may be something that we can always help with as well.
Yes, that's always good to know. Yes.
They are, yeah, they've been a great.
Sorry, I was going to say that actually could be particularly important for Dr. Grove's research because it's in this area of embryos. But again, that when she talks, you
they've been everybody here has been a great resource whenever me and Angela putting grants and helping us with feasibility and feedback. So I'm sure that everybody would be delighted to if we can help in any way with your research as well.
Okay, great. Thanks very much, then.
Thank you so much. Brilliant. Bye, everybody.
Bye-bye.
Everyone.
Cheers.